So hi everyone to our 10th webinar of IMMUNO-model cross-section. For those who are not familiar with our cross-section, this is the cross-section dedicated to modeling immunotherapy response and toxicity in cancer. We are a growing community. Now we are around 400 members mainly from European countries, but also like worldwide from United States and so on. So the aim of this IMMUNO-model cross-section is to foster research and innovation in the field of preclinical immuno-oncology models. And these webinars are like opportunity to discuss the field and also to connect with different stakeholders in the field of immunomodels. So today we have two speakers. I'm very happy that they found time and willingness to give very interesting presentations on natural killer cells in action from bench to bedside. So the first speaker is Professor Valés-Gómez. She's coming from Spanish National Research Council in Spain, in Madrid. And over the past 25 years, she has made pioneering contribution to natural killer biology and their role in cancer immunology. She's collaborating worldwide with many excellent scientists. She was also visiting researcher and professor in many established universities like Stanford and Harvard University. She's co-author of 85 peer-reviewed publications and holds multiple patents, exemplifying her commitment to bridging basic immunology with innovation. So the floor is yours, and we have then the second speaker, and at the end we have the questions and answer session. So please write your questions in the chat, or you will have the opportunity to ask live on Zoom. So Professor Valés-Gómez, please, the floor is yours. We don't hear you, just unmute yourself. Okay, great. Oh, not yet. I think the microphone is muted.
Okay.
Yeah, now it's working.
I couldn't find myself around the multiple screens. Okay, well, thank you, Barbara, for this very nice introduction. And thank you very much for organizing the session and for inviting me to present our work on natural killer cells. And also to the IMMUNO-model for having all this bubbling community. Can you see my pointer? Yeah.
Yes.
Can you see the pointer?
Yes, yes.
Okay, so yes, as you were saying, we are basic research laboratory working at the Spanish National Center for Biotechnology in Madrid. And we are interested on how NK cells can eliminate tumors and also how tumors try to evade this response against the immune response. So since probably some of the people in the audience are not very familiar with NK cells, today what I wanted to do is to introduce NK cells, how they were discovered, and the complexity of NK cells. And very importantly, take into account the timing of all these important discoveries so that we can understand why we are where we are with NK cell translation into the clinic. And importantly, also some of the concepts that the NK cell community introduced and were extended to other areas of immunology. And then after that, I will tell you about a very exciting project that we started trying to study one of the first immunotherapies ever used, that is the use of BCG, the tuberculosis vaccine, to treat bladder cancer. So if we start at I'm going to be back like 40 years or 50 years, but in science it's not such a long time. And the very first discovery that led to the identification of NK cells was around 1973-74, and it was a very annoying anti-tumor background that appeared in lymphocyte cultures, both in human and mice. And this was called natural killer activity. And it was strongly discussed for like a decade because many people, many immunologists, supported that it was some kind of T-cell culture in vitro artifact or some type of unconventional T-cell. So it required a lot of time to actually realize that it was a different cell population. And it was when the TCR work was started to be done that we could confirm that NK cells do not rearrange the TCR. So then it was definitely shown that it's a different cell type. And then at that time, already a hypothesis was proposed. That is the missing self hypothesis. And it's because the very early studies, both in human and mouse NK cells, clearly showed that unlike T cells, NK cells were not MHC restricted. But very importantly, they preferentially kill tumors that were lacking MHC class 1. And when do we lose MHC class 1? Well, one player is viral infection, the other player is tumors. So they were discovered in the context of tumor, and they are probably carrying about the same things that we need to eliminate viruses. And I think this is very important because the tools that we are going to use in both fields, in tumors and viruses, many times are the same. So if we take the picture of the 80s of an NK cell that is still the same, it was defined as a large granular lymphocyte because they are bigger than T cells. But what they have in particular is these granules that are preloaded with granzyme and perforin, so they are ready for action. And very soon, the main marker for human NK cells, that is CD56, was included in the picture, which is actually an addition molecule. It's also called NCAM. And today, I have to tell you, I'm going to focus on human NK cells because the murine NK cells are very similar in function. They use very tools that have evolved to be very similar. But the families of receptors, the molecular details are very different. So I'm going to focus on human NK cells today. The other thing that was very soon discovered is that many NK cells express this CD16, that is an Fc gamma receptor that can recognize opsonized targets. In the 90s, sorry, I didn't pass. In the 90s, we were still trying to understand how NK cells were working, and the missing self hypothesis had predicted that there would be some inhibitory receptors that would care for MHC class 1. And actually in 1995 was when the first inhibitory receptors in NK cells were cloned. And this was a complete change in paradigm because what then was discovered was the immunoreceptor tyrosine-based immunoreceptor tyrosine-based inhibitory motif, which is the counterpart of this item that was already known. And this is a signaling motif that is in charge of sending the inhibitory signaling after engaging the ligand. And at that time, it was kind of complicated to introduce this concept because most people were very familiar with engaging a receptor and becoming activated, but now we were engaging a receptor and actually sending a negative signal, which is something that nowadays we have very clear in our minds because of all the checkpoint inhibitors, and we already know about this balance of signaling. But this was the moment when people started to talk about balance of signaling between activating and inhibitory receptors. And I was lucky enough to be at that time in a lab that was caring for NK biology. So then KIRs, actually, which were called initially killer inhibitory receptor, changed the name to killer IG receptors because they come in pairs. You can have the activating and the inhibitory counterpart. And then very soon, we had an explosion of receptors on NK cells. And I told you in 1995, we didn't have any receptors. In 1997, we had 2 gene complexes in 2 different chromosomes having all kinds of different receptors, activating, inhibitory. Some were stochastically expressed on NK cells. Some of them were alleles of different genes. And at that time, there was this review, like from no receptors to too many. We had too many receptors, still no ligands for many of them. And it was very difficult to understand, to try to imagine how all these receptors would control the biology of NK cells. So we are on the verge of the 21st century, still trying to figure out what an NK cell can do. And in parallel, also, something that was very important for the NK field was the discovery of several factors that help proliferating and responding NK cells, which are the cytokines IL-12, IL-15, IL-18, IL-21. And if you compare what was happening in the T-cell field, that is like in the 80s, we had already our first trials with IL-2 and interleukin-2 activated lymphocytes. And it wasn't until 2018 that we had the first clinical trial with IL-15, because things were coming much later, and also the regulation to do clinical trials became more stringent. So it's not just that NK cell came later to the field of immunology, it's also that everything, some things could move very quickly, like the cloning of receptors, but other things require more time. And then what we had is the identification of ligands that came little by little already in this century. So, and then we started to know about how they develop in the body, and also something which was kind of revolutionary, that is that NK cells, although they are classified as the innate immune system, they can have some adaptive-like functions, like for example, they can respond in an antigen-specific manner to cytomegalovirus, for example. And we can also create some memory-like NK cells with some combinations of cytokines. So from the very beginning, we had the idea that NK cells could kill tumors. And then there was a first wave of clinical trials in the early 2000s where people were infusing in hematological stem cell transplant cells with a mismatch between MHC class 1 and KIR of the donor and the receptor. So, and these transplants resulted in lower rates of leukemia relapse, presumably due to the missing self-response by NK cells. And then much more recently, we started to play with NK cells in the clinic in the same way as CAR T cells, there were CAR NK cells produced, and with this memory like NK cells. And you probably will hear much more about NK in the clinic from the second talk, but in 2022, we had this review with summarizing some of the trials that started at the time. And what we can say about these trials is that we know that NK cells are very good for patients because they are very low in toxicity. They don't cause any neurotoxicity, no cytokine release syndrome, no graft versus host disease. So in general, they are very safe, but the work in progress needs to work on persistence and efficiency of the NK cells to kill tumors. And the latest that we know is that there is a lot of heterogeneity in NK cells. So we know that NK cells are able to kill tumors, that they probably evolved to eliminate viruses, but they use the same kind of tools. They have a lot of activating and inhibitory receptors that they need to balance to have their action. But they can be specific for certain targets. And all the different NK cells that we have in blood can have different functions. So we need to kind of choose or select the ones that are more important for cancer if we are interested on that. So in peripheral blood, NK cells are about 5 to 15% of our lymphocytes in a healthy donor. And they are also in very irrigated organs like liver, lungs, placenta, etc. And what we can find in blood is a majority of cells, and 90% of NK cells are this phenotype of CD56 dim. So that means that they have only a few CD56 molecules per cell, and they also express CD16, and they are considered the mature NK cells. And then the remaining 10% is CD56 bright, which has a lot of CD56 per cell in the cell surface, but they do not have so much CD16, and they have different cytokine receptors, etc. So these two types of NK cells have different functions. Our CD56 dim are the cytotoxic NK cells, so they have this activity of the granulation. They are the ones that are going to respond very quickly against a target cell, releasing granzyme and perforin. They also have other mechanisms to respond against tumors, like a trail receptor or fast ligand. And then, as I was mentioning with CD16, they can perform antibody-dependent cell cytotoxicity, that is also called ADCC. And then our CD56 bright are more regulatory. They are important producers of cytokines, and they respond also to cytokines in the surrounding media. And they are very important to crosstalk with other cell populations. They are mainly found in tissues, and in general, NK cells are thought to be there to communicate also and to make a bridge between the innate and the adaptive responses. And all these activities occur in different situations. So if we have a healthy cell, we would have what we call tolerance for NK cells. Usually it involves an inhibition with MHC class 1 and maybe not many stimulatory ligands. And then the NK cell wouldn't kill this target cell. And then there are other situations in which either MHC class 1 can be lost, and that's the missing self situation, or you have a very stressed cell that start to upregulate a lot of stress ligands like NKG2D ligands, and then you have what is called induced self. And of course, what I was mentioning about antibodies, if you have an opsonized target, then you will have antibody-dependent cytotoxicity. So all these different situations are responding to different ligands that are in the target cells. So if I tell you there are many different types of NK cells, and we need to find the one that is important for tumor elimination, I could show you this cartoon, and you can go kind of a bit mad, because you have all these different activating, inhibitory, chemokine receptor, adhesion receptors that can be combined in different cells and in different quantities, or you have different alleles. They are all germ-like encoded. But for now, maybe it's easier if I show you this picture where you have something that looks like it is the same, but it's not exactly the same. So we have a lot of NK cells that are in peripheral blood, but if we talk about the percentage of NK cells in blood, probably we are not meaning too much. What we need to know is what receptors are expressed in these NK cells. And then with this idea in mind is when we started to think about what are the important NK subtypes for cancer elimination, and can we generate these anti-tumor NK cells in vitro. So that's when we came to our model of bladder cancer. And for those of you that don't know, this is a system that is used in a type of bladder cancer. It used to be called superficial bladder cancer, but this is a bit misleading because these are very high risk tumors. They are very undifferentiated, and if they are left untreated, they can invade very quickly all the layers of the bladder. So these patients are treated directly into the bladder. They receive instillations of BCG, and like 60–70% of the patients are tumor-free 3 years after treatment. So it's a therapy that is very efficient and really revolutionized at the time bladder cancer treatment. And it's a treatment that the patients receive in several occasions. So usually they have an induction phase where they receive weekly instillations for a month and a half. Then they rest for like 2–3 months, and then they have the maintenance that comes like 2–3 instillations separated by rest periods of like 2 months or so. So because this was working so nicely, we were thinking, well, maybe we can learn from it and see how NK cells are working in this system. And so, well, we had 2 main questions. I'm going to talk today pretty much about how we approach how NK cells are working in the BCG model, but we also have another project that is also very interesting, which is we can take ex vivo samples of urine mainly from these patients, which is that has been in contact with the tumor, and we can look at in this urine for cytokines and cells that have been recruited there. So, and then we can probably see the patients that are responding, what do they have mainly, and help the ones that are not responding to check for biomarkers. But this is a story for another day. Today I'm going to tell you about how we started working with BCG in vitro mainly, and learn about how NK cells are being activated in these cultures. So what we did was take our peripheral blood mononuclear cells, and we did not eliminate adherent cells, myeloid cells, or anything, just take the whole PBMC and put them in day 0 with BCG. We didn't add any cytokines or any other factors, and let them in culture for 7 days. And then after these 7 days, what we wanted to know is how these NK cells are doing, what do they express, how they respond to tumors, etc. So what we published this a few years back, and what we could see is that on day 7, our NK cells have upregulated CD56, but this only means that they are activated. It's not like they are similar to the CD56 bright in blood, and I was introducing this so that we can see the difference, because these cells are actually very cytotoxic against tumors. They still express CD16, so they have the features of a mature NK cell with very high CD56, and they are able to perform very strongly cytotoxicity against tumors. So we look at the cytokines that were in this culture, and we could find some IL-12, TNF-alpha, interferon gamma. And then we started to think, well, can we keep these cells in culture? And what we looked at the beginning was that both in vitro and in the urine of patients, the cytokines were very dilute. We have very low concentrations of cytokines, but still our NK cells were activated and were able to kill tumors. So what we thought is that, well, if we take BCG to change the phenotype of NK cells, that we assume that is giving a particular phenotype and that leads to dysfunction, can we keep them in culture just with minimal concentrations of cytokines so that is sufficient for their proliferation and to keep them in culture, but to avoid exhaustion? Because if we keep NK cells with high amounts of cytokines for long term, then they usually become exhausted. So that's what we started to study, and we did some RNA-seq analysis, single cell, and we also did some titrations, and we came to the conclusion that we were going to use IL-12, IL-15, IL-21, and we left away IL-18 because the receptor was not including a lot of response in the BCG cultures, but also because IL-18 stimulates a lot NK cells, and we had the idea that if we put IL-18, they become too crazy too quickly, and they kill everything you are going to put in front of them. But then they become exhausted sooner. So that's the combination we decided to use, and we wanted to use really, really low concentrations of these cytokines and try to put them for longer times and see how they were performing. So for you to compare with what is used in other systems, which is, for example, for CAR-NK cells, many times there is IL-15 or IL-21 that expands these NK cells, or for cytokine-induced memory-like is 12, 15, and 21. So we are changing the composition and lowering the concentration. And for example, with cytokine-induced memory NK cells, what people do is they take IL-12, IL-15, and IL-18, they pre-activate overnight, then wash the cells, and then keep them with some low concentrations of IL-15 during the weeks. This concentration is still higher than the one we use. And then when you restimulate with cytokines or with cells, these NK cells are producing high amounts of interferon gamma. So we have some differences with this protocol. We have some differences too on the phenotype that is obtained with cytokine memory-like NK cells. And what we did was, as I was telling you, day 0, we have our BCG. Then we leave them for 7 days. And then on day 7 is when we put our low concentrations of IL-12, 15, and 21. Then we leave them for another week, stimulate again, and so on. We have gone up to 1 month. Probably we can fine-tune a bit more this protocol. We can probably do it at day 6, and then we can shorten this day 14, which is the first day that we would have our product ready to use. But anyway, before I talk you about any product, I'm going to tell you what they do. So when we have our NK cells produced, during the first week, if you look at the white circles here, what we have is the number of cells in the culture. So the first week we go down in number of cells a bit, and then they increase over the weeks. And more importantly, this black bar is showing you the percentage of NK cells in this culture. So without any need of cell sorting or selection or anything like that, we have like 50% of NK cells in 2 weeks. And in some donors, we can reach up to 80%. So we have a very good expansion of NK cells, but what is more important, they kill many different types of tumors. So here I want to remind you that our assay is done on day 7, just before we add the cytokines. So this culture has been almost left starving for a week, and then that's when we do our killing assays. So no restimulation before the killing assay. And then what we see is that they kill all type of tumors. Here we have examples of bladder, melanoma, breast, gastric, colon, lung, but we have done hematologic, we have done pancreas, we have done many other types. And these cells are very good killing different types of solid tumors. What the difference between our cells and the cytokine-induced NK cells is the production of cytokines. So if you take this protocol where you add the IL-15 during the week and so on, then you can see that the cells produce interferon gamma in high amounts. Our cells produce less interferon gamma. They produce some, but not so much. But what is really different is that the degranulation is much higher in the cells that have been treated with BCG than in the cells that have been treated only with cytokines. So basically, we have a system where we think we can produce NK cells. Well, we have characterized the phenotype, but I don't want to be boring with different names of receptors of NK cells that are very complicated. But we have a very particular phenotype of NK cells that we call Bill NK cells because they are BCG prime IL interleukin stimulated. Basically, we can enrich them a lot in culture. We can have them growing without having overstimulation and exhaustion. And we thought, well, this is maybe something that we could use as a new way of producing a cell therapy. The other thing that was very that looks very promising for us is the fact that we could stimulate cells that come from immunosuppressive environments. On the left, we have expanded NK cells from tumor patients, from blood from tumor patients. In the right, we had some bone marrow samples from patients where the NK cells were less than 1% in the starting culture. So we could expand some NK cells and make them kill. So all these things make us think that our system can work in vivo and could be a good idea for a new therapy. So basically, we would have the advantages of NK cells, which is the low toxicity and the promising results that have been already produced. And we think that we have overcome some of the challenges like the short-lived nature in vivo and the capacity of responding against solid tumors, which is still a challenge for many of the therapies nowadays. So then with these ideas, we have our next set of questions. So the first one is, can we really repurpose BCG to produce a novel cell immunotherapy? And then is like how these BCG stimulated NK cells are going to work in vivo, and can they infiltrate tumors? Like now we need to respond to the difficult question, right? So for this, we are doing preclinical experiments with several collaborators, which is great because it's what we really need in order to continue in the orthotopic human bladder cancer. And then we are also doing currently a model with NSG mice osteosarcoma in collaboration with the group of Antonio Pérez. And we will do later a leukemia model. Still in my lab, we are working on characterizing better these immune cells. It looks like the myeloid cells are very important to help growing NK cells or some subtypes to actually avoid the growth of NK cells, so that we can play a bit better on the scalability and expansion of these cells, which is probably our next step. And also, I would like to finish by telling you that in my lab, beside the BCG project, we have other projects going on. We have projects working with NKG2D as a model for recognizing tumors by NK cells. This is a receptor that is very important to recognize tumors to recognize tumors by NK cells. And we have been playing a lot with the biology of the ligands of NKG2D. We showed that one way of evading the response is by secreting these ligands in extracellular vesicles. So we had another field that is also that we study a lot in the lab, that is how to characterize proteins on extracellular vesicles. So in collaboration with a Spanish company, we have developed a method to visualize markers on extracellular vesicles, and especially on biological samples, we have gone down to like 12 microliters and be able to visualize the extracellular vesicles in plasma or in urine. And using this technology during the COVID pandemic, we use this method to develop serological tests to know which patients had been responding to SARS-CoV-2. So, and now the more important slide is the people that actually had done the work. I have really motivated people in my lab that have been working very hard to take all these projects forward. Also to my collaborators, this is they are really important to keep working for us, and to the funding agencies. And I don't want to conclude without telling you that we are organizing this symposium, this workshop with IMMUNO-model for March next year. So please save this date because it's going to be great. We have confirmed international speakers that are really very prominent in the NK field. But what is very important is that we are going to have roundtables to look at regulatory aspects, how to transfer your results to the clinic and to industry. And I think it's going to be a very nice way of working together to learn and to collaborate. So thank you very much for your attention. And now we will listen to Antonio and we can talk about this a bit later. Thank you very much.
Many thanks. It's a very impressive work and congratulations. So now I will give a word to Professor Rosalinda Sorrentino. So she will introduce the second speaker. So Rosalinda, please, the floor is yours.
Hi, good afternoon. My name is Rosalinda, and it's a great pleasure for me to introduce you Dr. Antonio Pérez Martínez. He's the head of the Pediatric Hemato-Oncology services at the University Hospital La Paz, and is a full professor at the University of Madrid. And I can say that he's a key person in NK studies, and he has done so much work. He has a multidisciplinary team of specialists. So I would like to introduce you Professor Antonio Pérez and leave him the floor.
Thank you so much for your kind introduction. First of all, let me know if I did. Can you see the slides properly?
So now it's
Yes.
not in full presentation mode. It's like before, but it's also fine. So.
Can you see now my slides?
Maybe freeze them. Okay.
Oh yeah, now it's perfect. Great, thanks.
Perfect. Okay, thank you so much for the kind information and introduction. First of all, I would like to apologize for my voice. Some virus infection happening from last Friday, and this is not my normal voice, so I'll do my best to try to explain my next talk. So thank you, Mar, for your outstanding presentation regarding the basic of the NK cells. I'll try to do the clinical point of view, how we use in the clinics NK cells. First of all, let me introduce myself. These are going to be the 6 topics I'm going to try to address. First of all, I am a pediatrician, so I'm going to talk to you about pediatric condition. I'm an expert on bone marrow transplant and cell therapy, so this is how we are going to address the NK cell therapy. Because, in pediatric setting, we need for innovative therapies. We are working for the last 2 decades to try to bring to the clinic the adoptive NK cell therapy. So at the beginning, with the missing cell hypothesis, this is something that Mar already talked to us. But in addition, when this missing cell hypothesis is not working properly because we don't have this key HLA mismatch setting between donor and recipient, how can we overcome these inhibitory signals with, for example, this IL-15 ex vivo cytokine approach? So at the beginning, let me introduce myself. This is my trip in medicine. I've been learning from the best pediatrician in the world in bone marrow transplant, and first in Spain, then in the US, Germany, again to the US and UK. So all of these people are experts on bone marrow transplant field in kids, and they bring the adoptive cell therapy into the clinic. In the last 1 decade, I'm working in Hospital La Paz. I'm also professor of pediatrics, and I have this very nice research institute where we are trying to do some research on NK cells and try to bring to the clinic as doing translational clinic and translational research project. So at the beginning, as I said, bone marrow transplant and cell therapy, to me, as the best platform we can address these NK cells. And how? Well, as you know, we have been very worried the last decades to try to eliminate graft versus host disease from the bone marrow transplant setting. So we have different ex vivo platforms to try to eliminate T cells from the graft. T cells are responsible of the GVHD, and try to retain and to maintain the graft versus leukemia effect mediated by NK cells. So in our actual strategy, we are using in leukemia kids patients, we are using reduced intensity conditioning, and we are using cells as a strategy to improve graft versus leukemia effect without inducing or with a mild GVHD. This is what we are trying to do in our setting, and this is how is our first 1 year of treatment. We are using cells, and you can see we're using NK cells from the beginning of the transplant at day 7. So with this approach, we have now very nice data that we have very nice overall survival and disease-free survival in the last 5 years of our protocol. So how are we going to do this point? Well, as you know, one of the main points in pediatric cancer is we have the mandatory to increase the cure rate and the quality of survivors for children and adolescents with cancer. So one of these, the decalogue of these 7 points, one of them is innovative therapies. These innovative therapies, to me, one of them is adoptive NK cell therapy. And we have been working, as you can see, from the 2011 and previous in some clinical trials using, for example, NK cells in solid cancer using hematopoietic stem cell transplant. We have some experience also in trial using NK cells in refractory acute leukemia, AML and ALL. We also have experience in NK cells as adoptive cell therapy as consolidation strategy to control relapse in AML. And now we also have some experience of NK cells in solid tumors out of transplantation. This is a trial is already going in sarcoma, and I try to explain this trial, PINK, that we have already finished. And I will try to explain you how we are the results of this trial. So we have a lot of experience in the clinical setting. We have many first in human case adoptive therapy in kids. And at the end of the study, we see that most of these patients have a response. However, the response, we cannot able to keep this response in a prolonged time of period. So at the end, more than 70% of the patient response have any overall rate respond to NK cells, but a few of them we are able to took. So because of that topic, we first of all, in the haplo stem cell transplantation setting, we first try to analyze this missing cell hypothesis. The missing cell hypothesis was described by the Italian group, by Andrea Belardi, in AML adult patients in haploidentical stem cell transplantation setting. It was very important to the field of bone marrow transplant because it makes transplant easiest. It was easiest to find a donor for everyone. We don't need a matched donor. And even when we have haplodonor, the graft-versus leukemia effect mediated by NK cell could be better. How does it work? It works because when you have a missing HLA ligand from a receptor for a clear inhibitory receptor, for example, when you have HLA-C group 2 missing and you have the receptor in the donor, you have an alloreactive NK cell. So you can, at the end, you can have 3 different alloreactive NK cells, depends on the missing ligand and the presence of the NK cell repertoire. Making a little a small summary of that, so we have a recipient without the ligand, we have a donor with the ligand, so this is the kill ligand mismatch hypothesis. And with that approach, if we are able to obtain that, we have seen that there is an improvement in the outcome after leukemia, after transplantation, when we use this kind of models. In the pediatric setting, the evolution of this kill ligand model was a little more complex. Wing Leung and Rupert Gretinger, they also said that we need the missing cell on the receptor, the presence of the ligand on the donor, but in addition, we need the repertoire, the receptor in the donor that is able to recognize the missing ligand. So this is why this is called the kill receptor ligand mismatch model, is the Memphis model. And with that approach, we have not only find an impact in AML, but also in some ALL, for example, in infant ALL. So at the end of the history, we are able to find that of all NK cells, only the NK cell that they are able to express 2DL1, they are able to recognize the missing cell of the C2 ligand, or kill 2DL2, DL3 are able to recognize the missing cell of the C1, or the 3DL1 are able to recognize the BW4. So applying all of this knowledge to the donor selection, we can, as you can see, improve the outcome of the mismatch hematopoietic stem cell transplant in different projects, not only in the US, but also in some European countries. So because of that, this is the work I'm going to present it today. It's an example of how adoptive NK cells in the clinic we can use when IL-15 NK cells can overcome inhibitory signals. So this is the paper we published this month. So and the background is that, as you can see, that high-risk pediatric acute leukemia, when they relapse or they are refractory, they need a stem cell transplant. Match-related or unrated donor is the first option. However, haploidentical makes transplant more universal, more faster, and now we can do it even increasing the graft-versus leukemia effect. So how can we do graft-versus leukemia effect without using EBSD? Is something that we can try to incorporate in this haplo setting, the power of the NK cells. So NK cells, as you know, is a subset of the lymphocytes in the peripheral blood. It's about 15%. We have different subtypes. We have different signals, inhibitory or activatory signals. And at the end, the NK cell can eliminate abnormal cells that they are infected by virus or they are transformed by NK cells without need of a prior sensibilization, using the missing cell or using the inducing cell hypothesis. When the missing cell, you can see the absence of the inhibitory ligand for the key inhibitory receptor, or the inducing cell when you are able to express ligands for the activatory receptor. Because of that, and this NK cell can be educated, and we can analyze that KIT2DL1 are able to recognize HLC2, and they are tolerant for the autologous setting, but they are going to be very active in the receptor ligand model for the patient cell when there is a missing cell in the C2 and the presence of the KIT2DL1 in the donor cell. This NK cell alloreactivity is something that is going to decrease the relapse rate, the viral infection, and improve at the end immune reconstitution, graft-versus leukemia effect, and the outcome after transplant. So because of that, we try to use alloreactive model. However, 50% or 55% of donor-recipient pair, they are not biological mismatch. So can we do something to try to overcome this inhibitory signals in KIR-HLA match setting? So, as you know, IL-15 is a very important cytokine for NK cells. They are like they are involved. This protein is involved in the survival and the maturation. They can increase the expression of activating the receptor, including interferon gamma, TNF alpha. And the most important thing, NK cells are not going to be senescent, they are not going to be exhaustive. So there are different strategies to try to put IL-15 in this model. As you know, IL-15 in vivo is a little toxic. So most of the cases we try to use ex vivo IL-15 NK cells, and the IL-15 we are using is the ex vivo overnight stimulation, and then we wash all the and we remove the IL-15 from the cells, and we just infuse these NK cells stimulated overnight in this case. So we ran a trial in adolescents, young adults, and kids in Spain. We started, it takes 4 years to complete 18 patients. You know, ALL, AML is high risk. Now we have very little patients, so we need a national study. First of all, we use 3 different cohorts until 20 million per kilo of NK cells, 20 to 50,000 cells per kilo, or more than 5 times to the 7 to 1 time to the 8 cells per kilo in 2 different settings. Allo NK cell when donor recipient was KIR-HLA mismatch, or ex vivo IL-15 NK cell when donor and recipient were KIR-HLA match. So we obtain the cells by these multi devices, you know, Kinimas Plus. We stimulate in the hood, and then in the if they are if the cells were KIR-HLA mismatch or recipient, we just infuse the cells at day 7. But if the donor recipient were KIR-HLA match, at the time we go into a GMP facility, we stimulate with IL-15, and then we infuse as well at the same time. So we did some different time points with clinical variables, outcomes, cytokine profile, NK cell infusion, immune reconstitution, and all of these things. So at the end, we were able to obtain 18 patients, 9 in each arms. They all were very high-risk patients. Most of them have received a previous transplantations. And as you can see, we were able to obtain close to 80% of survival at 1 year after overall survival, disease-free survival, and graft relapse-free survival was about 60% in both cohorts. We have not found any difference between overall survival, disease-free survival in alloreactive setting or IL-15 setting. So this is now good news, because we were able to control or to pull the NK cell at the same time as alloreactive NK cells. However, our IL-15 NK cell group, we have a more incidence of acute GVHD, grade 1 and grade 2. You can see between 50% and 11%. You know, when we analyze the cytokine profile, we found that at 1 month, the IL-2, the IL-13, the IL-17 was higher in the IL-15 NK cell cohort. Also, there was a very interesting increase 1 year after transplant. So we found an early cytokine profile stimulation and a late cytokine stimulation. We don't know, remember that NK cell were only infusion during the day 7. When we analyze the cytokine profile, the activating and inhibitory receptor, IL-15 stimulating NK cell have more stimulating phenotype, NK46, NKG2D, CD69. So some of these activatory markers also were able to degranulate higher and more cytotoxicity than non-IL-15 stimulated NK cells. However, immune reconstitution in alloreactive NK cell setting and IL-15 non-alloreactive setting was similar, and we have a very robust data on that with a very early NK cell, anti-cell, and CD4 and CD8 recovery. And in the alloreactive setting, as you can see, that the KIR2DL1 NK cell were the faster to recover, with a peak in the KIR2DL2 that they decreased later, and the KIR3DL1 that they were most setting. But as you can see, the alloreactive clones are increasing during the time in the alloreactive KIR setting. However, in both cases, in KIR-HLA mismatch and KIR-HLA match, we observed that NKG2D at 3 months was more higher in the IL-15 expression. So that means that the NK cell clones KIR mismatch that they are not able to find in the alloreactive NK cells, we can observe the activatory signal in the KIR-HLA match setting. So probably doing the same role as the alloreactive KIR inhibitory NK cell we're doing in the KIR mismatch setting. So at the end, we observe that NKG2D is a very important activating receptor to control, we hypothesize, to control the relapse rate. And we have not found any relapse in this cohort of group during the first year after transplant. When we correlate some markers with HGBD, chronic GBD, or transplant-related mortality, overall survival, we found, in addition, that CD69, it was predicted very nicely the group of patients that can have a worse outcome. It means they have more transplant-related complications and mortality. So at the end, we found a model of that IL-15 NK cells infusion were able to control the graft leukemia effect in a setting where NK cell should be energy, because they are expressing, the leukemia cell are expressing the HLA class 1. So we can overcome this strategy with these IL-15 NK cells at the beginning. Now, this is our conclusion of this topic. We consider that IL-15 stimulated NK cells in KIR-HLA match haplotransplant is safe and effective, comparable to the same infusion of allo NK cells in KIR-HLA mismatch. The maximum tolerated doses we were able to obtain was 5 × 10⁷ NK cells per kilo. They have the similar overall survival, the CT survival, transplant-related mortality, and relapse rate with allo NK cells. However, IL-15 NK cells have higher expression of activating receptor and enhanced cytotoxic capacity. In addition, the pro-inflammatory profile may contribute to higher risk of GVHD and TRM, while potentially can enhance the graft leukemia effect. So at this time, NK cells show similar expression and functional patterns in both groups, suggesting comparable immune reconstitution, and CD69 could be a biomarker to try to monitor and try to do something to control the transplant mortality. So we are now designing the second cohort of this PIN study in order to compare IL-15 in KIR-HLA match, compare IL-15 NK cells without NK cells. It tried to be now not equality study, just effectiveness study. And also, we are trying to use these markers as a strategy to control increasing the immunosuppression if needed, when the 69 is higher early at the first month after transplant, to increase immunosuppression. So that's, as Mark said, the most important slide. Just to want to thank all the doctors in the clinic, the researchers in the lab. Thank you, all of you, for your attention. I would like to highlight Carmen Mestre, who was the PhD study of this PINK trial in my lab. So thank you so much. I'm happy to have any questions.
Okay, so many thanks for your impressive work. So really congratulations. So now we can start with the discussion and answer session. So we have already some questions in the chat. But if somebody wants to ask in person, please raise hand electronically, and then you can have a word. So I see here one question. Amazing talks. Thanks. What do you think about the use of NK92 cells as a therapy, and how critical is interleukin in support for their in vivo efficacy, considering their IL-2 dependence in vitro? So this is a question from Paula Martin Rubio, and I think it's a question for both of you.
Well, first of all, thank you so much, Paula, for the interesting question. I have no experience at all with NK92 as adoptive cell therapy. I know there are many trials. The main advantage of NK92 is off-the-shelf product. You can use faster, and you can. But as Paula said, they need the IL-2 to expand into the patient. So all the trial, when we use IL-2, we are inducing in the NK cell more senescence phenotype. So this is why most of the studies are trying to move to improve the persistence and to have a more powerful and more persistent effect of the NK cells. We are trying to modulate the response using IL-15 or other cytokine as IL-15, IL-18, IL-21, and this memo-like phenotype that Mark suggested in his previous talk. But it's a very good question. I'm sorry I cannot have any data on my experience, but I know that there are many papers, and to me the best advantage of this approach is the NK cells are already with you, just off the shelf. And this IL-15 NK cell, you have to think for the donor. All of these things make a little things more complicated.
So, Mark, do you want to add something to that?
Well, I don't have experience with therapy directly, but yeah, what Antonio was saying is that people are now engineering NK92 with IL-15 or IL-21 so that they can self-proliferate without the need of exogenous IL-2 or any other exogenous interleukin. So these are the things. The other worry that some people have with NK92 is that at the end it's a tumor cell line. So they have the advantage of being off the shelf, but it's also in itself came from a tumor. So some people don't like this idea, but it's something that is being tried in different settings. Yeah.
Okay, thank you. So I have one question. So what about this, now it's very hot topic, this microbiome. So how is microbiome connected to NK cells? We know that some bacteria can stimulate the activation and so on. So what's your, from the basic research and also clinical view of microbiota and use of antibiotics in combination and so on with the therapy? So that's my question.
So, well, the microbiota, if you have a healthy microbiota, the NK cells have learned to become tolerant to it. So I guess if you have an infection, NK cells are very important to kill infections. They are more important with viruses than with bacterial infection, but probably if you have a bacterial infection, you have an imbalance of what you are having on the surface of your cell. You can have stimulating ligands for NK cells. I'm pretty sure that people are looking at this quite actively.
Yeah.
I think it's very important in the clinical setting. One of the things we are seeing is how important is the conditioning for transplant. There are some conditioning that makes the gut, very toxic to the gut. And this inflammation into the gut makes, in a mismatch transplant, even with reduced intensity conditioning, makes a lot of signals to the T cells and to the NK cell that can induce GBSD. And this could be an explanation why we have this IL-15 phenotype activity in NK cells and more gut GBSD. Our conditioning, most of our patients use melphalan, and melphalan is very toxic into the gut. We use melphalan because we have problems in most of the country, most of the center in Spain, we cannot monitor busulfan. Busulfan is more toxic to the liver, but it's less toxic to the gut. So probably in one of the things we have to try to address, if we move to a less gut toxicity conditioning, instead of melphalan, use busulfan, probably the signals to the microbiome and to the gut and to the NK cells can induce less GBSD and can leave the NK cells only to make the job in the target on tumor or leukemia cells, not on this on target of tumor cells that is in the GBSD. So this is an hypothesis. So
okay, thank you. So I see a hand from Isozinum, so sorry for the wrong pronunciation. So I see a hand, you can ask a question.
Yes, so good morning, everyone. So first, thank you for your nice presentation. I have a question for the first speaker. I'm most interested on to understand the mechanism by which the BCG bacteria can lead to NK cells. So I want to understand, is it possible that this PBMC, like having the antigen presenting cells, can take the bacteria and produce some interleukin like IL-12 or 15 and try for the production of NK cells? Is it possible by this mechanism?
Yeah, there is for sure the contribution of some presenting cells. I mean, we have been looking with fluorescent BCG what cells are binding the BCG, and we can see that they are CD14 positives, the one that are binding BCG. The thing is, we don't know if it's because of the presentation or if in general. What we assume is that there is some processing of the bacteria by the dendritic cells and myeloid cells, and then this is producing IL-12. We have interferon gamma, we have TNF-alpha in the culture. We have all these cytokines that make NK cells proliferate. So if we remove completely the myeloid cells, the activation of NK cells is not complete. We are now trying to find which one of the myeloid population is beneficial or is not beneficial, because we think we have the two two types. We have some myeloid cells that are helping, some that are not helping.
Okay, thank you. Thank you for your answer.
Okay, thank you.
And can also, like a last question, he was talking about tolerance phenomenon by the natural killer cells. Can you please tell me about like specifically how these natural killer cells can induce a tolerance?
Sorry, I didn't get the question. Is it for me or for?
I mean, can you hear me?
Yes, we can hear you. It's just sometimes it's a bit, lost. But, yeah, please repeat the question. Yeah.
I just want to understand by which mechanism the natural killer cells can, in organism, distinguish like self-antigens to, let's say, foreign antigen, like a mechanism of tolerance.
Okay, so the mechanism of tolerance comes a lot with these receptors that look for MHC class 1. So there are different specificities of HLA-C that are recognized by particular receptors. So that's self versus non-self. And then, for example, in the case of the BCG stimulation, the tumors are recognized via NKG2D, which is an activating ligand that recognizes some molecules that are related to MHC class 1, but they are not MHC class 1. They are called MIC-A, MIC-B, and ULBPs. And these are molecules that are not present in healthy cells, but when the cell becomes stressed, and one of the stress is tumor transformation, these ligands go to the cell surface. So there are, in general, most of the ligands that are recognized by NK cells are not expressed in healthy cells, because otherwise you would be killing your own healthy cells. And NK cells have this array of receptors that are there to survey if you have some infected cell, tumor transform, or any kind of stress cell.
Okay, thank you.
May I ask a question? Do you know, just similarly to what was the previous question, do you know more about the NK cell exhaustion in this NK cell therapy? Can you give us some detail? And also, if I can do the second question, I also saw in Professor Gomez's presentation that when you did the single cell RNA sequencing of primed NK cells, you also had STING upregulated, if I remember well. Do you think that that pathway could also be important for NK, especially because it could be activated after the chemotherapy or other type of cytotoxic therapies?
So about the STING pathway, yeah, we were thinking that maybe some interferon, type 1 interferon could be playing a role. And we have actually been playing a lot with interferon alpha and beta. But what we have seen is that once you have the BCG, adding interferon alpha or beta doesn't help anymore. It's like with the BCG by itself is doing this kind of changes in phenotype and activation that is enough on itself to have the NK cells working nicely. And if we add then the interferon, it doesn't help. The same way, if we add the cytokines earlier on in the week, it doesn't help. So it's like, it's not like we are producing a type 1 response kind of with the BCG. Something, I don't know, it's some different phenotype. I don't know if it's looking not too scientific the way I'm saying it, but it's like it doesn't, we don't need to use the STING agonists or anything in order to improve it. And we have not found a way to avoid the use of BCG and substitute it by the STING agonist. And about the exhaustion, I don't know if this was for Antonio, right?
Both of you, if you can.
Well, in our therapy, your question about the NK cell exhaustion markers, or if we were able to monitor the NK cell, as you know, it was those we cannot a marker to follow, just follow the chimerin, so because they are allogenic for the donor. So we follow most of the NK cell all after transplant are from the donors. So we observe in the kinetic a peak at the beginning in the mismatch setting of the KIR2DL1 and in the IL-15 NK cell cohort, the NKG2D, and we haven't seen any other senescent biomarker, just activation marker 69, but the PD1, LAC3, all of these things, we haven't seen any additional. For sure, we don't think that NK cell can persist from day 7 until 1 year. So probably what we are seeing in this first wave and the following wave it comes from two ways. First of all, the NK cell that we put in the graft, and we put at day 7, and second, the NK cell that recover from the bone marrow transplant. It takes more time, and in this high inflammatory cytokine environment, they can do like a bystander effect that probably makes this second wave. But at the end of the picture, we don't know exactly what is the mechanism underlying this effect. What we have able to check that no one relapsed during the first year. So this was amazing, because the relapse rate, and you know that in haplo is the major problem in high-risk leukemia. The major failure of bone marrow transplant is leukemia relapse. Probably we are able to keep on this high inflammatory NK cell from the beginning until the end. We decrease as much as we can the chance of the leukemia relapse. Now, because this trial was comparing the positive control group, who are alloreactive NK cells, comparing with IL-15 NK cell. Now we are going to, and it was equal. Now we are trying to do a trial comparing the superiority of the IL-15 NK cells in the match setting, comparing with the no NK cells, or probably with not stimulated NK cells in the match setting. So at that time, we want to see an improvement on the outcome using the IL-15 NK cells in this outcome setting prospectively. I mean, if you see this data in historical cohort, cohort from US, from Italian group, when you use match KIR-HLA match NK cells, the outcome is worse. But this is retrospective data and historical data. We now want to compare prospectively these IL-15 NK cells against NK cell, non-alloreactive NK cell in KIR-HLA match setting, just to see if we can see an improvement that hopefully we can see.
Thank you.
Okay, any other question from the audience? Everything clear? And okay. So if no further questions, we can conclude, I think. So thank you both of the speakers. I think it's amazing work, and it's, I think, growing interest in NK cells, which I think they deserve the attention. And they can be nice also, how to say, parallel with also CAR T and T cell therapies and other therapies. So very exciting field.